Hi, today we are going to discuss about one more important essay question from lipid metabolism. The topic is fatty acid synthesis. This is otherwise known as linen spiral. Okay, we have two types of fatty acids, that is saturated and unsaturated. In unsaturated, polyunsaturated and monounsaturated fatty acids. These saturated fatty acid and monounsaturated fatty acids are synthesized in our body, whereas polyunsaturated fatty acid is not synthesized in our body. That is why they are known as essential fatty acids. Usually the fatty acids are stored in our body as triacylglycerol. Next one is, what is the significance of these fatty acids? We all know the synthesis of hormones, synthesis of bile acids, synthesis of vitamin D, and synthesis of some metabolic cellular components like phospholipids. These are all synthesized from fatty acids only. Like one or two lines you have to mention about the significance of fatty acids. Third one is how it is synthesized. We have two ways, that is two pathways for synthesis of fatty acids. One is de novo synthesis pathway, another one is elongation of fatty acids. De novo synthesis means newly synthesized fatty acid. Elongation means from that fatty acid we are going to add 2 carbon atoms. Finally, we will get 16 carbon palmitic acid like long chain fatty acids. So two ways means de novo synthesis is one type, another one is elongation of fatty acids. Next one is, what is the site and location? Cytosol only. The organs are liver, kidney, brain, adipose tissue and lactating mammary gland. This is actually known as extra mitochondrial lipid metabolic pathway, otherwise known as cytosolic pathway. Next one is, you have to know for any, you should know about the initial product and the end product for any pathway. Then only it will be easy for writing the essay question. The initial product is 2 carbon acetyl CoA, end product is 16 carbon palmitic CoA. That is the concept of this lipogenesis is, initially you will get only 2 carbon molecule. This 2 carbon molecule add up on 2 carbon molecules, and finally you will get 16 carbon length of fatty acids. That is lipogenesis or fatty acid synthesis. Next one is, what are all the requirements for this pathway, fatty acid synthesis pathway? One is acetyl CoA, another one is NADPH. NADPH. Acetyl CoA, we all know it comes from the aerobic glycolysis. The end product of glycolysis is pyruvate. This pyruvate is converted into acetyl CoA with the help of pyruvate dehydrogenase. Actually, we already uploaded one more separate video for PDH, that is pyruvate dehydrogenase, because it is your 5-mark short notes. Already we have uploaded. At your free time, you all of you go through the PDH. With the help of PDH only, acetyl CoA is synthesized. Actually, this is from mitochondria. Whereas NADPH, the source of NADPH is another important MGR University question short notes, right? So we have three areas or three sources for NADPH synthesis. Number one is HMP shunt. First two reactions or two steps of HMP shunt produces two molecules of NADPH, right? Second one is malic enzyme, which is present in the cytosol. The role of malic enzyme is conversion of malate into pyruvate. That is, one molecule of NADP plus malate produces NADPH and pyruvate with the help of malic enzyme. Third one is TCA cycle. In TCA cycle, we are having important one enzyme, isocitrate dehydrogenase, which is the synthesis of alpha ketoglutarate from isocitrate. Right? What is the role of isocitrate dehydrogenase? Production of alpha ketoglutarate from isocitrate. In this isocitrate dehydrogenase, we have two isoforms. One is cytosolic isocitrate dehydrogenase, another one is mitochondrial. This cytosolic isocitrate dehydrogenase produces NADPH, which is useful for the fatty acid synthesis. Next one is three phases of it, that is three reaction or three steps for this lipogenesis pathway is very important. We have divided into three phases. First one is, acetyl CoA actually produced in the mitochondria. Cytosolic lipogenesis occurs in cytosol. So from mitochondria to cytosol, how this acetyl CoA is transported, that is the first phase of lipogenesis. The shuttle name is oxaloacetate to malate conversion, or malate to oxaloacetate shuttle for the transport of acetyl CoA from mitochondria to cytosol. Number one. Second phase is after entering into the cytosol, this acetyl-CoA converted into malonyl-CoA. This is actually first reaction of your fatty acid synthesis pathway, carboxylation reaction. Very, very important example for your biotin vitamin carboxylation reaction. Third one is usually for all the other reactions, one enzyme involved for the conversion of A to B like product. Whereas in fatty acid synthesis, we are having complex of enzymes. That is one acyl carrier protein which is attached with the 6 continuous enzymes. All are doing separate activity, which will be very useful for the immediate reaction takes place in fatty acid synthesis. That is fatty acid synthase complex, which consists of condensing unit, reducing, that is reduction unit, and finally releasing unit. Releasing unit means release of palmitic acid, 16 carbon fatty acid, right? Next slide we are going to discuss about these three phases in detail. Okay. Yes. Now we are going to discuss about the three phases in detail for the lipogenesis or fatty acid synthesis. Okay. The first one is malate oxaloacetate shuttle pathway. Here what is the role of this shuttle is, we are having acetyl-CoA in the mitochondria only. We have to shift the acetyl-CoA from mitochondria to cytosol. This is the role played by this shuttle. That is, malate is converted into oxaloacetate, oxaloacetate is converted into citrate, vice versa, citrate is converted into oxaloacetate. The oxaloacetate is converted into malate because acetyl-CoA and oxaloacetate is not permeable to the membrane, whereas citrate and malate is freely enters into the membrane. That is the reason for the conversion of these compounds, right? So in the mitochondria, we are having one enzyme citrate synthase. With the help of the citrate synthase, oxaloacetate is combined with acetyl-CoA and produces citrate. This citrate is freely enters into membrane from the mitochondria to cytosol. Whereas in cytosol, we have another enzyme ATP citrate lyase. This citrate is lysed and produces oxaloacetate and acetyl-CoA. This acetyl-CoA enters into the lipogenic pathway, that is fatty acid synthesis pathway. This oxaloacetate is converted into malate in cytoplasm, which is again enters into the membrane. This is the concept for first phase, that is malate oxaloacetate shuttle pathway, right? Second one is the first step of fatty acid synthesis pathway is conversion of acetyl-CoA into malonyl-CoA, right? Here, this is a carboxylation reaction. That is why one molecule of CO2 is needed. Another one is the enzyme, regulatory enzyme name is acetyl-CoA carboxylase. This is otherwise known as rate limiting enzyme. What is the meaning of rate limiting enzyme? If you are having adequate amount of palmitic acid, that is end product is okay, means the pathway will be automatically switched off. If there is essential or need of palmitic acid, automatically this is switched on. So biotin vitamin is essential for this carboxylation reaction. This is the first step of lipogenic pathway. That is why we need one molecule of energy compound, that is ATP. So these are all the basic steps for the conversion of acetyl-CoA into malonyl-CoA, right? After this malonyl-CoA enters into cytosol, how it will produce palmitic acid? We have 5 important steps. One is condensation, another one is second is reduction, hydration, one more reduction, finally thioesterase. These all 5 reactions are done by this fatty acid synthase complex. This is multi-enzyme complex 1. 2 monomers, so dimers, 1 dimer, then 3 domains, 3 units, condensing unit, releasing unit and reduction unit, and finally 1 plus 6, that is 1 acyl carrier protein plus 6 enzymes, totally 7 enzymes, which are the components of fatty acid synthase complex, finally releases palmitic acid, right? Then coming to the after any pathway, the important point for the next point is regulation, how it is regulated. Number one is substrate availability. If the excess carbohydrate is available for lipogenesis, excess carbohydrate is converted into glycogen in liver. Apart from this, excess glucose molecules converted into lipogenesis, right? If there is no excess glucose means there will be no place for lipogenesis. Availability of the substrate, we have two compounds which regulates. That is, one is stimulated, another one is inhibited. The stimulator is citrate and the inhibitor is palmitoyl-CoA. That is, it's nothing but if the palmitoyl-CoA is adequately present means automatically it will inhibit the pathway. If there is need of palmitic acid, citrate will stimulate the pathway. Right. Second one is acetyl-CoA carboxylase. I already mentioned rate limiting enzyme or regulatory enzyme of fatty acid synthesis. Third one is hormone regulation. We have two groups. One is insulin, another one is glucagon and epinephrine. Insulin always stimulates the lipogenesis, whereas glucagon and epinephrine inhibit the gluconeogenesis. That is why insulin is a hormone for lipogenesis, whereas glucagon and epinephrine are the hormones for lipolysis by phosphorylation and dephosphorylation. Next one is, for any pathway, last step is very, very important. Here the last step is butyryl ACP. ACP means acyl carrier protein. With the help of thioesterase releases palmitic acid. Right? Again, this acyl CoA enters into 7 cycles, produces, that is elongation of 2 carbon atoms with the acetyl CoA 2 carbon atoms. So finally, you will get 16 carbon long chain fatty acids, palmitic acid. Here one more MCQ point is, palmitic acid is synthesized only in liver and adipose tissue, whereas capric acid and lauric acid produced from lactating mammary gland. This is very, very important MCQ. Do we have any disorders in fatty acid synthesis? Yes, we have two disorders. One is dicarboxylic aciduria, another one is zellweger syndrome. These two are very, very important short notes. What is the name of the enzyme defect? What are the clinical manifestations? How you are going to investigate this disease? And what are all the treatment you have to apply for the disease? These are all the important points for this disorders of fatty acid synthesis. Okay.